I think we can start now. And well, first of all, good morning to everybody. On behalf of the Spanish group of Crystallography and Crystal Ground, I wish to welcome you to this new series of webinar, Ciencia en la Red, Science in the Net, that begin today. Now I will say a few words in Spanish, but the rest of the session will be in English. Simplemente quería de nuevo daros la bienvenida a esta serie de conferencias que hemos programado, Ciencia en la Red, para este curso, programadas por el grupo especializado de cristalografía y crecimiento cristalino. Estas conferencias las vamos a celebrar durante todo el curso, en principio los terceros martes de cada mes a las 12:30, en principio, salvo aquellas que no puedan ser por distintas cuestiones. Los títulos y los conferenciantes ya los comunicaremos convenientemente en su momento por las vías habituales. Bueno, recuerdo también, sobre todo a los jóvenes, que pueden solicitar certificados de participación en estas conferencias. También quería decir simplemente que el grupo de cristalografía y crecimiento cristalino, además de esta actividad que ya tenemos programada, al mes que viene, en noviembre, vamos a hacer una sesión presencial en Madrid, en el Instituto Rocasolano, donde será una sesión científica que además aprovecharemos para entregar diversos premios que no hemos podido entregarlos en estos 2 años que hemos pasado en las circunstancias que todos conocemos. Así, en esta sesión, que será el día 17 de noviembre, se entregarán las medallas, las insignias de oro del grupo a los profesores María Isabela Riortúa, Miguel Ángel Cuevas y Enrique Gutiérrez. Además, también entregaremos el premio Javier Solans, que entregamos todos los años a un investigador joven. Entonces, quería animaros a asistir, o bien presencialmente, también lo vamos a retransmitir a esta sesión, que será la primera presencial desde hace prácticamente 2 años. En esta sesión también daremos los resultados de las elecciones parciales que se están celebrando en este momento. Os invito a participar en ambas cosas, por una parte en las elecciones y por otra parte en esta sesión presencial. Por otra parte, también quiero animar a los miembros del grupo G3C a que, si tienen alguna propuesta de nuevas actividades, etcétera, nos lo comuniquen a la Junta, que estaremos encantados de implementar aquellas ideas que partan de nuestros miembros del grupo G3C. Sin más, y agradeciendo de nuevo vuestra presencia, voy a dar paso a la conferencia para la que nos hemos reunido. Now, Professor Lourdes Infante will introduce to you our speaker, Dr. Francesca Stanzione. Thank you very much, Francesca, for accepting our invitation. And Lourdes.
Okay, thank you, Pilar. And first of all, thank you all the audience to be here, and to Francesca to accept our invitation to participate in this webinar. Francesca Stanzione is a computational scientist who is working at the Cambridge Crystallographic Data Center. She is working in drug design, specifically working in the development and improvement or optimizing the tools to do docking, to do drug design. And for me, it's a pleasure to attend her conference today. I hope we can learn a lot from her. And I'm not going to say anything else, just very enthusiastic about listening to you. Thank you very much, Francesca.
Thank you very much, Luz, for the kind introduction. Thanks everybody for joining the webinar. It's a pleasure to be here. And today I will focus my talk on about molecular docking in drug discovery. And I will try to provide you an overview of the molecular docking, how it works, and how it can be a powerful tool in drug discovery. So if I can go to the next slide. Okay. So here I'll show a very well-known drug discovery pipeline. And we all know that drug discovery is a challenging process, and identifying the right lead compound is a determining factor of the overall success of the project. In 2016, the Tufts Center of the Study of Drug Development estimated that the cost associated with the developing and bringing a new molecule to market has increased by almost 145% in the last decade. Furthermore, while the average time to bring the drug to a clinical trial has decreases, the success of drugs that are able to obtain the FDA approval has dropped to 12%. So therefore, you can see there is a clear that discovery pipeline needs to be optimized. So computer-aided drug design, or so-named computational tools, has helped to reduce the costs and the time associated with the drug discovery by directing experimental research towards optimal compound more quickly. So here are listed a few of the computational tools that can be used in drug discovery. Of course, I'm not going in all of them, but I will focus on molecular docking, and I will provide kind of few examples of how docking could be implemented, could be worked with the molecular dynamics combined. So within the computational tools, techniques such molecular docking and virtual screening have provided a valuable complement to the time-consuming and expensive experimental process of high-throughput screening. So the ability of computational screen a library of compounds that either possess similarity toward a known or ligands, or a complementarity towards target structures, have proven to be successful and identifying highly focused subset from which active that can be isolated and then experimentally confirmed. So to date, generating such educated guesses have provided many examples of lead compounds, and several marketing drugs and clinical candidates are known to have been discovered or optimized with the aid of computational tools, including molecular docking. So molecular docking is, as I mentioned, is the most common computational method. This has been widely used since developed in the early 80s. So it requires 3D coordinates of protein and the ligands, and involves two basic steps. So prediction of the ligand conformations, as well as its position and orientation in the binding sites. This is defined as the pose of the ligand, and then the assessment of the quality of the pose that is defined as a score. So a further task of the docking procedure will be to score active compounds higher than known inactive. However, this level of accuracy is difficult to achieve because there are many other factors involved that are external to the protein. So therefore, molecular docking only aims to get the prediction of the ligand pose and the assessment of the quality of the pose in a correct way. So, why molecular docking? Molecular docking applications in
Beatriz.
Yeah. There is a problem with the sound.
Yeah, I can see.
Francesca?
Francesca?
Maybe it's a problem from Francesca, I think. Maybe she should go out
A problem
and
here.
get it again. Yeah. Okay.
Francesca, are you there? Sorry.
Este tipo de tips, este tipo de que estamos por aquí, que cualquier cosa que tenga por chula, pues
I suppose it's a problem of
para contar
the Wi-Fi or
entender un poco la vida como este tema de
okay.
Sorry, everybody. Probably Francesca is trying to reconnect again. In a few minutes, she will.
es un poco muy visual para mostrar un poco de ese juego, cómo funciona, pero si tenéis algún vídeo de estos que mole con alguna escena de estas de batalla, de fusiones, de lo que sea, pero me interesa para ponerlo. Sí.
Beatriz.
Sí.
Francesca está desconectada.
Está desconectada, sí.
Sí. Imagino que está intentando conectarse de nuevo. Creo que no depende de nosotros,
No,
sino de ella.
de ella.
Sí, sí.
Sí. Le ha escrito un email, me ha contestado. No, me ha contestado una respuesta automática.
Sí.
Sorry, of course, there are technical issues as usual. So they are making works on close by my house. So my internet just dropped.
Don't worry, Francesca.
Don't worry.
Apologies.
2 minutes, no problem.
Okay. I don't know where you lost me.
You were just in this slides, I think so. Starting this slides because you passed. Can you put the previous one, because I think you finished the other one and you passed us to this slides. Yes, that's correct. When you started talking on here. Yeah.
Okay, perfect. So apologies again. I hope it's not happening anymore. But
Don't
okay.
worry.
So I was here. So why molecular docking? So molecular docking in drug discovery are varied. So we have been has been applied in several ways. However, knowing exactly how and where the ligand binds helps structure activity studies to rationalize the design, so the changes that you want to make to the ligand in order to optimize protein ligand interaction, to improve activity, and to avoid changes that will result in clashes between the protein and the ligand. Docking can also be used to facilitate predictions from mutagenesis studies by providing binding site hypotheses and to assist the crystallographers in fitting of substrates and inhibitors to electron density map. Docking has proved to be extremely successful. Therefore, it has been developed and improved for many, many years. So over the last 2 decades, more than 60 different docking tools have been developed in academic and commercial settings. So there are several available reviews that detail the evaluation and the comparison of different docking programs. So I will not mention these tools, but in the slide here, you can find one of the reviews that talk about the comparison of the tools. So the sampling process to find the ligand poses in a docking is not trivial. The conformational space involves many degrees of freedom, including the rotation and translation of molecules relative to each other, the additional conformational degree of freedom of both the ligand and the protein, and sometimes degree of freedom related to the solvent. So in practice, with the current computational research, it is impossible to explore such a large space within a sensible time scale. So hence, efficient sampling of conformational space is still a challenge in docking, and some compromise has to be used. So therefore, most of the docking programs account for while limiting the flexibility in the protein to the region of receptor that interact with the ligand, or keeping the entire protein as rigid. So the underlying reason of this is that since the ligands are usually small molecules, they are likely to undergo a larger conformational changes. Furthermore, and given the small size, accounting for ligand flexibility is also computationally affordable. So how the pose is found, so how the search is performed. So in docking, the ligand and the protein binding sites are represented as donor acceptor pharmacophore points and hydrophobic pharmacophore points with a different radii. So these pharmacophore points are named fitting points. So the searching algorithm tried to match the protein and the ligand fitting points by matching donor with acceptor and hydrophobic with hydrophobic cavities. So in case of unacceptable orientation, so for example, clashes between the protein and the residues in the binding site, the ligand is then reorientated until acceptable orientation are obtained. So when the pose is obtained, so then the quality of the poses has to be evaluated with what is defined, what is named scoring function. So scoring functions are fast, approximate mathematical methods that use to predict the strength of interaction between two or more molecules. So 4 aspects of the scoring function should be considered when assessing the reliability of a scoring function. First, the scoring power, that is the ability to reproduce scores which linearly correlate with the experimental binding affinity data, if these ones are available. Ranking power, that is the ability to correctly rank a given set of ligands that bind to a common target protein by their binding affinity. Again, when these binding affinity are known. Then we have docking power, that is the ability to identify the nothing binding pose of a ligand as the one with the best score, and when screening a large set of generating decoy poses. And lastly, the screening power, that is the ability to identify the true binders to a given target protein among a library of random molecules. So ideally, an accurate scoring function will be performed equally well on all these 4 tasks. However, such scoring function does not exist yet, and each existing scoring function only performs well on one or two of them at the same time. So here I'm providing a high-level overview of scoring functions, just providing a few information about which scoring function are available. So those they are distinguished in two classes. That is classical and machine learn-based scoring function. The classical scoring function are further grouped in three main classes: the physics-based, empirical, and knowledge-based scoring function. So the main difference is in the way they treat the energy terms and they are derived. So classical scoring functions are based on the assumption that the change in free energy upon binding of a ligand to its target can be decomposed into a sum of the individual energy contribution, and that all these energy contributions are linearly combined. So in reality, such linear correlation may not always exist. And I will say that the two major limitations in the classical scoring function are the minimal description of the protein flexibility and implicit treatment of the solvent. So machine learning based scoring function instead, they use a more sophisticated techniques such as random forest or support vector machine and deep learning to approximate not linear problems. So, however, they are more computationally expensive than the classical one. So over the years, several studies have assessed and compared the different scoring functions. Each one has its virtues and drawbacks, and none of the scoring function available outperforms the others in all the tasks that I mentioned before. But each scoring function may perform better than other in a specific task. So machine learning scoring function, instead, they have been proved to outperform a classical scoring function in such tasks as ranking and screening. However, they are rarely directly incorporated in the docking software and are mostly used for rescoring. So after this overview, I told you that all the scoring functions perform similarly. How do you know which scoring function you use for a specific system? So one way to evaluate which scoring function to use is by experimenting and optimizing the choice based on active, inactive, or decoy test sets for each specific target protein. So this strategy, of course, is subject to the availability of experimental information on active ligands. So the customized, how it works is that the customized active and decoys or inactive test set are used to assess the performance of each scoring function in order to find the best one available for the specific target for a virtual screening campaign. So when you don't have enough data, so you don't have enough active ligands, scoring functions are usually selected based on the ability to reproduce the native ligand binding conformation, or what is named self-docking. Success of such prediction is defined by the root mean square deviation value between the top ranked ligand conformation and the experimentally observed structure. Generally, if the root mean square deviation is 1.5 angstrom or below, the prediction is considered successful. So to compensate for the drawbacks of individual scoring function, the simultaneous use of different scoring function or even individual terms of multiple scoring function has been widely used to obtain what is called consensus score. So one way to perform consensus scoring is to reevaluate the best docked pose of each compound with another scoring function. Only the top scored compounds common to each scoring function that has been used will be identified as a candidate for the bioassay. So consensus scoring is thought to increase hit rates either by reducing the number of the false positive or by statistically reducing the error in the scores or in the ranks. So how it works. So here I'm providing a very brief description of how to set a docking experiment. So the first step is to relate to the selection of protein and ligand to use. So a docking protocol, as I mentioned before, requires the 3D coordinates of both protein and the ligands. So the major resource of 3D structure of macromolecules is the protein data bank, while the ligand structures, there are several actually databases that provide the ligand structures, and to mention a few of them, there is Zinc, Campbell, or even the Cambridge structure database. When the 3D structure of the target is not available, protein structure prediction techniques such as homology modeling are commonly used to obtain the 3D model of target protein. So homology modeling relies on the correlation between the sequence of a target protein and its homologous protein structure available in the PDB. However, with the structure predicted by AlphaFold, you can see that it's much easier to obtain 3D guesses or 3D coordinates of protein nowadays. So if the 3D structure of the ligand cannot be obtained experimentally, they can be instead generated using software that are able to convert 1, 2, and 2D structures like SMILES and SMARTs in three-dimensional molecular structures. So there are several several tools available from academics and industrial providers. The second step required before running a docking is the preparation of protein and ligand. So first, for the protein, for example, if there are missing atoms or missing residues, these need to be added to the protein, and this is particularly relevant if there are missing residues or missing atoms into the binding sites. Then we need to add hydrogen to both protein and the ligand. So the protonation, I want to highlight, that is a key step when working with crystal structures and play an important role in the prediction of the correct binding mode. For example, as shown here, it is important to check the correct protonation states of the imidazo ring of histidine that in the neutral form can be protonated at either one of the 2 hydrogen, while in the protonation state, in the charged form, both nitrogen are protonated. So histidine is also a weak base with a pKa of 6, and its protonation state is slightly affected by the surrounding environment. So as for the protein, so we have to protonate the ligand as well. And for ligand, it's important to set the proper tautomers, protomer as stereoisomer, because these different entities may result in alternative binding modes that can affect the efficiency of docking and then the virtual screening campaign. The third step is the definition of the binding site. So the binding sites are usually determined experimentally. So if there are, for example, for a complex or protein ligand complex. However, if in absence of such complex, the binding site can be predicted with several tools. So programmatic cavity detection is, I would say, is a well-researched field, and many methods are available using different algorithms are available from in academic and by industrial provider. So another step is the definition of structure of water, or what is called active water. This is important because water molecule can coordinate hydrogen bonds between the protein and the ligand, and therefore they can affect the binding pose of the ligand itself. So, however, hydrogen bond network may be ligand specific. So, for example, this could be a problem when you are doing a virtual screening campaign. So for this reason, when active water molecules are included, they should be ideally treated as flexible. So only after all these steps are performed, the docking run can start. And the searching algorithm will explore the conformational space to find the pose, and then the scoring function will rank the candidate solution based on the energetic parameters that I showed you before. So when the docking is performed, then there is the evaluation step. So where you want to evaluate the quality of the docking solution. So for example, one way of doing this is to check in the hydrogen bond pattern. So to check if all hydrogen donor and acceptor in the ligand are satisfied, or if the 3D structure of the protein ligand complex is available, if it's possible, if the binding pose, so the conformation pose of the ligand is equal to the one of the crystallographic structure. So are we able to reproduce the native conformational pose of the ligand? So docking is currently in a mature stage, so and also in a mature stage of development, but is still far from being perfect. Most docking programs available are not able to predict known binding poses with a success rate between the 70 and 80%. However, the calculation of accurate binding energies is one of the major limitations in a docking, in molecular docking. And this directly correlated with the approximation on protein and water. So on the poor treatment of the protein flexibility and the treatment of the solvent. Such limitation can widely affect more sophisticated docking approaches, such blind docking, where there is no definition of binding sites and the ligand is docked to all the surface of the protein. And the covalent docking, where the ligand is covalently bound to the protein, and to reverse docking, where the ligand is docked against an array of protein. So particular in the challenges, there are several challenges in the blind docking. And again, blind docking refers to docking a ligand on the whole surface of the protein without knowledge, prior knowledge of the target pocket, so the specific binding site. In blind docking, the entire protein is considered as the region where the ligand might bind, leading to a much large conformational space, and therefore in a running time. So they are very computationally expensive. Moreover, the complexity of blind docking grows exponentially according to the possible binding sites that are available in the protein, which severely limits its use in practice. However, blind docking methods which can predict bound conformation without a prior knowledge of the binding site location will be needed for a full automated computational approach for in silico drug design. So one of the ways, so the space, the enormous space, search space is a primarily problem in docking, and there is where the research is working on trying to tackle this problem. So there are two possible ways to mitigate this, either to reduce the search complexity and split the docking boxes in multiple boxes, sacrificing, however, the flexibility of some part of the ligand. Another way is to repeat the search several times using different seeds and then merge the result together. And instead of running a one single large blind docking. So another alternative approach is to decrease such the computational complexity of blind docking is to combine binding site prediction tools to identify possible ligand binding sites with the docking experiment, followed by fine tuning and ranking of the initial solution using scoring function and some optimization methods. So the other challenge is covalent docking. So historically, so this is a new challenge because historically drug discovery mainly focused on non-covalent drugs due to the potential off-target effects and toxicity issue of irreversible covalent drugs. However, in the recent years, and actually with the outbreak of COVID-19, we have witnessed the resurgence of covalent drugs. So the rational design of covalent ligand is still faced with the particular challenges, mostly related to the fact that covalent bond formation, bond breaking, and bond rearrangement are quantum mechanical phenomena, which cannot be handled or adequately handled by the force field or empirical approach that are typically used for non-covalent protein ligand interaction. However, existing docking tools have been adapted to be working with covalent ligands, particularly when the nucleophilic target is known. So if there are prior knowledge of the protein and the binding site and the nucleophilic target in the binding site, traditional method can be used, traditional docking tools can be used. And in such case, the docking task is just to elucidate the binding mode and to run compounds according to their suitability to fit in the active sites after covalent ligand to the protein. For advanced tasks, such as the design of covalent ligands and breaking and forming bond, there are more sophisticated approaches, and these are usually applied when there are not much prior knowledge of the system. So for example, a QM-based scoring function has been developed to describe covalent binding of ligand, but this is still to be optimized, and there are still research studies ongoing to optimize the covalent docking. Another challenge in docking is what is called reverse docking. So as the name suggests, this involves docking of a set of one of few ligands against a pool of protein molecules that could be protein families, for example, with the aim of identifying a potential target, their binding affinity, or polypharmacology profile. So reverse docking can provide a valuable contribution in the drug repositioning or repurposing and in the treatment of infectious disease, including, again, COVID-19. And therefore, the tool has become more and more popular with the database of provided predicted structure provided by AlphaFold. Research docking also contribute to rationalized designing for less toxic and multi-targeted drugs. And however, the main limitation is primarily related to the occurrence of the current scoring function to distinguish between the true target from no target protein. So this is because many, if not all, the contemporary scoring functions are designed for docking or screening small molecule in the protein binding site without special optimization for reverse docking. However, several tools have been, several researchers and several papers have been published on evaluating the different scoring function in the reverse docking. And I would say that most of these scoring functions can be applied to the problem. So where to use molecular docking? So despite the limitation and the challenges that I mentioned before, docking has proved to be extremely successful in drug discovery and drug design. Therefore, it has been developed and improved for many years. Molecular docking is widely used in drug discovery for rationalized ligand activity toward a target of interest and for virtual screening campaign. So, but beside these applications that were the one that the docking was designed for, docking can also be used for target fishing to identify a series of targets for which the ligand presents a good complementarity, or for the prediction of adverse reaction or off-target interaction based on the complementarity between the ligand and protein. Moreover, docking is also currently employed for identification of ligand that simultaneously bind to a pool of selected target of interest. So like, as I mentioned before, for reverse docking, and these can provide information about the polypharmacology. And another use of docking is on identifying novel use of chemical compound with already optimized safety profile. So the drug repositioning and the drug repurposing. So despite molecular docking was originally designed to be performed between a small molecule and the protein, in the last decades, there have been a growing interest in other fields like protein-protein docking, protein-peptide docking, and nucleic acid docking. So protein-protein docking is an emerging research topic due to its potential for predicting protein-protein interaction. So although protein-protein docking share the principle of protein small molecule docking, sampling of the conformational space in protein-protein docking is extremely challenging because of the large size of the binding sites and also not well characterized binding sites. So despite these, the complexity of the problem, a variety of docking methods are currently available for predicting the structure of protein-protein complexes. So the choice of the methods depends on the nature of the problem and the knowledge that you have of your system. For example, rigid docking, so the one that I described before, are generally used when the crystallographic structure of the protein to dock or a very close homologous and the complex of the two proteins are available. But when the crystallographic structure of the complex is not known, there are also several approaches that can be used, for example, using an ensemble of structure or a simplified protein representation, what is called coarse-grained. So another new field is the protein peptide docking that again is becoming popular since peptide drugs are gaining attention in drug discovery as a solution for target what are called undruggable intracellular protein-protein interfaces that are characterized by a large and relatively featureless interface. Protein docking, protein peptide docking involves the computational steps such as conformational sampling, structure refinement, the scoring that are similar to the one used for small molecule and so for protein small molecule docking techniques. However, proteins are more flexible than small molecule, and so they tend to adopt different conformation and a large number of different conformation, I would say. So over the past decades, a wide range of docking methods have been developed that can be used directly or indirectly on the docking peptides, and with actually a very decrease of success. So this method includes protein small molecule docking tools, protein-protein docking tools, and protein peptide docking tools. So the protein small molecule docking approaches are currently limited to a short peptide, so below 15 amino acids, but with a well-structured conformation. While protein-protein docking methods have been used for larger peptides with a well structure, with a more stable conformation, and while for more challenging peptides, they are still developing. They've been developed the more specific protocol that tackle the problem of flexibility of the peptides. Another development of the docking is nucleic acid docking. This again is becoming very popular because nucleic acid are potential drug targets for a number of diseases, particularly in the area of anti-cancer, antibacterial, and antivirus therapy. Generally, most of the protein ligand docking programs can be used to dock small molecule into nucleic acids, as they follow the similar principle and the similar physiochemical binding principle. However, while protein usually contain a well-defined and generally hydrophobic cavity, nucleic acids are characterized by more solvent-exposed binding pocket with a high charge density and polarity. Therefore, modification to existing protein ligand docking programs are required, for example, for dealing with the water that are they play a really key factor in nucleic acids structure. So another approach to use in docking nucleic acids is to use an ensemble docking. So using a set of predetermined nucleic acid conformation. So this can be derived by the X-ray structure, so available in the PDB, or by NMR model, or by molecular dynamics. So additional scoring function has been also developed in order to specific target the problem and the challenges in nucleic acid ligand affinity, and to address things like high polarity of nucleic acids. So the other way the docking have been evolved is the modality of by which the docking is used to assist the different tasks of drug discovery. So this modality has been changed over the time and with development of the tools. And although it was initially developed and used as a standalone method, docking now is mostly employed in combination with other computational approaches and within integrated workflow to overcome some of the most relevant intrinsic limitations that characterize molecular docking. So here you can see a few of those computational approaches. And according to the literature and to the available information, different in silico approaches can be combined with docking to generate integrated workflow that improve the prediction performances of the docking. For example, docking has been used in tandem with the ligand-based binding free energy calculation and machine learning approach to improve the prediction performances in the de novo virtual screening, and as well to assist target fishing, adverse drug reaction prediction, and drug repurposing. So likewise, different approaches can be applied at the different phases of the screening workflow to improve the dog prediction. For example, molecular dynamics could be combined with the AI-based method to identify suitable receptor conformation for docking, and then a ligand-based approach could be applied to rescoring the predicted docking poses. So you can see that there is a very large combination of tools that can be performed, and considering the number of in silico tools and techniques currently available, there are still countless opportunities for docking to be explored and integrated workflow with the aim to generate a single in silico workflow for drug discovery. So I think I'm done. So I am open to take any questions, and I hope you enjoy the presentation. Thank you very much.
Thank you very much, Francesca, for your nice presentation. Now it's time for questions, and all the attendants can hang up with the option you have down in your screen, or also can do your questions in the chat. There's only one comment on the chat. It says, incredible presentation. Thank you.
Thank you very much.
And there is a question from Armando Albert. I don't know if you can read or I read for you, Francesca.
I can read it. So Armando Albert is asking, what are the research lines and the CCDC to improve GOLD? So actually we were discussing before. So GOLD is a very popular tool and is widely validated, but we are still working on it. So in the recent year, we have put GOLD on a cloud and allowed to run GOLD on a cluster. So we have been working more about the efficiency of the tools and make it possible to screen a large library of compound. Recently, we have been working on allowing gold to run to 1D to 3D. So in a way that if you don't have the crystal structure of the ligand, that will be a protocol to convert SMILE to 3D. Mm-hmm. And about the scoring function, we are also working on optimizing and creating a new scoring function in gold that can contribute on problems like the protein peptides docking and nucleic acid docking. So there is still much to do, and we are still working on make it more available and suitable for all the new challenges in drug discovery.
There is another question from Jose Gavira. Can you read, Francesca? Will I read for you?
Yes, I can read. So I'm reading loud. So thank you. So he's thanking me for the interesting and illustrative talk. Thank you. So the question is, do you have any statistic number of the successful application of docking methodology to produce final marketed drugs? Example, percentage of the market, etc. So I don't have this number with me, and I don't remember, but I can. There are several reviews that talk about the marketed drugs that have been discovered by docking or a combination of docking with other computational tools. So imatinib is one that is coming to my mind that was just discovered by computational tools. But unfortunately, I don't have this number with me. So, but if you Google, it's pretty easy. You can find an exhaustive list there.
Mm-hmm. There is another question from Armando Albert, and he's asking, can we implement our knowledge on a particular target on the scoring function?
Yes. Okay. The knowledge is what is called the knowledge-based scoring function. So some of the scoring function has been developed on a pool of protein. So in this case, so there are also scoring functions that have been optimized for a specific target protein, or maybe you don't need to optimize the scoring function, but what you need is to optimize the protocol of the docking per se. So not just the scoring function, but for example, how well you want to explore the conformational space, what is the size of the binding sites, and so on. So all the protocol docking is very powerful also for this, because you can actually use your knowledge, what you know about your target, to guide your docking, your experiment. I hope I answer your question.
Thank you, Francesca. There is another question from Jorge Lozano. He says, hi, Francesca, nice talk. I am curious about the reliability of reverse docking. Could you comment on that? And which software application could you use?
Okay, that is a very, very challenging thing. So I am not an expert in reverse docking, first of all. Recently is a very recent thing, and we are still learning about it. But the reliability depends of the target. If there are similar target, and you are using similar target, you can use a specific protocol so that will work better. Or there are several challenges in the definition of the binding sites. So because there has to be similar, so how you define the binding sites, so you have to be large enough in order to explore. So there are several challenges. About the reliability of the tools, I cannot very comment on this, but there are several papers that actually have been proved the tool to work. And they have been used, for example, in this kind of drug repurposing, but there is actually very recent computational tools on COVID-19. So where they're trying to do, it's called is mostly based on repurposing, but they were actually improved using the reverse docking approach, how molecule can tackle COVID within other protein, for example. So that could be useful as well. But again, so it's not my field of expertise, I would say, but it's important to, as usual, when you work with a pool of compounds, it's important to have the correct starting point. So, for example, the database or the array of protein that you want to use for the docking experiment.
Jorge also asked you if there is a software linking reverse docking with AlphaFold.
As far as I know, not, but there are several research research ongoing. So now that AlphaFold is available to everybody, you can also see that there are several publications that are coming out using a predicted structure coming from AlphaFold. So this is kind of interest for us as well. So we will be working on things, and but the link is clear. So thinking about that, AlphaFold has solved the 93% of the unsolved protein. There is a word that has been opened. So that is a kind of high opportunity for reverse docking and trying to find the ligand. So repurposing, for example. So how can we use a well-studied molecule to solve other problems and to interact with other proteins that are just now being resolved. But again, those are predicted. So there are always kind of attention that you have to put on it. So even if it's very successful as AlphaFold, I don't think it's the end of crystallography of protein, I would say.
Okay, Jorge said, thanks a lot, Francesca, super interesting. And there is another question for Juan Ángel Organero. He's asking, what about docking of organometallic compounds with DNA or proteins? There are always problems on assigning charges to metal atoms. Is there any improvement in this area?
Yeah, I agree. So DNA and ions is one thing. It's a little bit more challenging. But metal, so there is, I can comment about gold, for example. So gold handles pretty well metals because it's able to assign, so it's generating the structure, so the interaction of the metal with the protein using templates that are available. And if the template is not available, you can build your own. So in this case, so in case of gold, I would say that it works good, very well with metals, but of course, depend on your system, and some more complicated things should be addressed. So about the improvement in the area, so I don't know, to be honest, if there is, I know that this is still actual problem, but I don't know how effort has been put in just protein and metal, but I think that will be some, let's say, development is more required for the DNA metal interaction, I would say.
I think that there is no more question. And then again, I would like to say thank you for your interesting talk and the last discussion. I would like to see more about the goal and the last development from the CCDC. And also if there is, I have a question about, is there any intention of trying to link or join molecular dynamics with goal in this case?
It is very interesting because we are actually working on defining what we are going with all the discovery tools. And one of the things that I am very passionate about is molecular dynamics. So it's my field of expertise. And we are trying to find a way to actually integrate molecular dynamics with gold. So for example, running a molecular dynamic under the hood to generate the conformation of the ligand or of the protein, or when you have from the docking, just running in molecular dynamics. So that is something that we would like to do. And so the answer is yes, that is something that will be very, we realize useful and interesting to do, just to make a nice workflow of the research topic and the research project.
Yeah, completely agree. And thank you very much, Francesca, for your talk. Thank you, everyone. Thank you to all the attendants to this nice webinar. And I think I'm going to finish or pass the talk to Pilar. Probably Pilar wants to say something about the next talk. Thank you, everybody.
Hello. I only want to thank Francesca other time for your very nice conference, and well, the other conference we have not in this moment completely concerted. Then we announce in time.
Okay.
Bien, muchas gracias a todos por participar. Las próximas conferencias las anunciaremos en su momento, y de momento lo que está ya programado es la reunión del día 17 en Madrid, en el Rocasolano, a la que me he referido antes. Por tanto, muchísimas gracias a todos y cerramos aquí este seminario.
Thank you, Pilar.
Gracias.
Gracias.
Thank you very much.
Thank you, Francesca.